Let's take a look at how hormones get a cell to do something. So in other words, what happens when the hormone binds to its receptor? How do we communicate the message? Way back in chapter 6, I told you to strictly separate water-soluble and lipid-soluble messengers. The lipid-soluble ones that are the ones that are going to the nucleus and become transcription factors, and we'll get those in a moment. And here first we're going to start out here this time with the water-soluble messengers. So they are either amino acid based, they're whole or bigger proteins even. So they are water-soluble and they cannot cross the phospholipid bilayer. So they can't just diffuse to here. They have to bind to a cell surface receptor. And I told you there are different options here for this. My number one favorite is always the G protein coupled receptor, and it should be your number one favorite too, because that's the one that's in physiology. That's probably the most common scenario. So here, let's start with that one. Here's your hormone, and it's binding to a G protein coupled receptor. And so the receptor here then is linked to this G protein, a heterotrimeric G protein. And once the hormone binds to the receptor, then something happens, the conformational change occurs, and something happens to the G protein to where the G protein then usually sends off a part of itself, either the alpha subunit or the beta gamma subunit. They travel together. And whatever is its next door neighbor, that one that it bumps into as a next door neighbor, that's how it carries on the signal. So if the G protein sends off either alpha or beta gamma subunit and it happens to bump into an ion channel, well, then it could open or close that ion channel and then change the membrane potential and then carry the signal from there. Through a change in membrane potential, for example, or then open other ion channels or release calcium or whatnot. So it's different options. Now, the other thing that can happen with a G protein is that it activates an enzyme. And so here they just put amplifier enzyme. Remember we talked about the adenylate cyclase being a very common one, or guanylate cyclase, or phospholipase C was another big option. So in the case we have adenylate cyclase, then we're making cyclic AMP as a second messenger. In the case where we had phospholipase C, then you would make IP3 and diacylglycerol as your second messenger. So there's different options, and if you forgot all about it, please review that from chapter 6. But at any rate, so here you have an amplifier enzyme, and remember, an enzyme doesn't just make one product, it makes sometimes 100 or even 1,000 products per second. So we have a big signal amplification step here if we're activating an enzyme that makes a second messenger. And then, of course, with the second messenger, there's different options. A lot of times we're activating kinases now, so like protein kinase A, something else, or we're releasing calcium in the case of IP3, if that was the second messenger. Either way, a lot of times we're having at least some phosphorylation steps here in these next steps, and then we activate proteins that got phosphorylated, and then we get some cellular response either from the cytoplasm or through gene expression changes. And here, the last option here, this would be a typical kind of growth factor receptor, or the insulin receptor actually looks like this too. So the hormone binds to this type of receptor. It has a single transmembrane domain, and the intracellular portion here that reaches into the intracellular side of the receptor is an enzyme. It's a tyrosine kinase, and tyrosine kinases, well, they're kinases that phosphorylate other stuff, and so we're kind of merging here with this pathway. Either way, make sure that you review that, how these water-soluble messengers, how they can get the signal across. And remember, they never enter the cell. They are unable to enter the cell. So they just bind to a cell surface receptor, and then the receptor carries the message into the interior of the cell, and we get that cellular response. I have an example here for you. This is my favorite example, I guess. And that will be here. We have a G protein coupled receptor. We have an amino acid based messenger. So here a derivative of catecholamine and or epinephrine, and that one binds here to this G protein coupled receptor. And well, the receptor is coupled to the G protein here with its 3 subunits, alpha, beta, gamma subunit. In this case, the alpha subunit shoots off as soon as the norepinephrine binds to the receptor. the alpha subunit from the G protein shoots off and bumps into its next door neighbor, which happens to be adenylate cyclase right here. The adenylate cyclase is an enzyme, as you can see, and it will take ATP and make cyclic AMP out of it. Now, the cyclic AMP, this stuff here, is a very, very popular second messenger in cells. And cyclic AMP will activate protein kinases, and once these protein kinases are active, then they will phosphorylate proteins, and these phosphorylated proteins that phosphorylate other proteins, and you have these whole kinase pathways, ultimately leading to a cellular response. Another thing that can happen is that a kinase will open an ion channel, which changes the membrane potential. This happens, for example, in the beta adrenergic receptors in the heart. There, the norepinephrine binds to a G protein coupled receptor, the beta adrenergic receptor in this case, and then indirectly opens an ion channel, which changes the membrane potential and causes the next heartbeat, the next depolarization wave to come earlier. And so that's how we're going to increase the heart rate when you have a fight and flight response. Okay, so that's the water soluble one. So make sure that you review that if you forgot all about it, and get up to speed. Now, the lipid soluble ones, I told you back in chapter 6, they're much easier because you don't need a cell surface receptor, you don't have second messengers and all that other stuff involved. There, you don't have the signal amplification effect built in either, but you have typically a more sustained, kind of prolonged response. And let's take a look at them. They're a little bit easier, I think, to understand. So in general, the steroid hormones, they're derived from cholesterol. That means they have this cholesterol, this steroid backbone, and they can be easily made with cholesterol, which we have no shortage of in our body. And so they're lipid soluble or lipophilic. They love lipids, and therefore they can easily cross the phospholipid bilayer of biological membranes. That means they can cross the phospholipid bilayer of the plasma membrane as well as the nuclear envelope, which is 2 phospholipid bilayers. They typically are carried by some protein carrier in the blood. They typically have a pretty long half-life, and they get across the cytoplasm and into the nucleus. Sometimes they need a cytoplasmic little carrier protein to help them across. But as I said, they have no problem crossing the phospholipid bilayers of the nuclear envelope, and then eventually they bind to nuclear receptors. I know it says here cytoplasmic or nuclear receptors, but here this is what you need to focus on, nuclear receptors. So ultimately, all of the steroid hormones, they act in the nucleus and they become transcription factors once they bind to their nuclear receptors. So basically, they activate or repress, so turning on or off certain genes, and that leads to the cellular response. So here are some examples of steroid hormones. Here you have the mother of all of those steroid hormones. That's cholesterol, and then you can make all the different other steroid hormones. Here you have estrogen, cortisol, aldosterone. So they're all related substances, and they all have that steroid backbone, as you can see here. So it's in pink. Let's take a look at the mechanism of action of these steroid hormones, or anything that works like a steroid hormone. So it's all the steroid hormones plus thyroid hormone works like this too. Okay, all the steroids plus one that doesn't look like a steroid but acts like a steroid hormone, and that would be thyroid hormone. So those lipid-soluble messengers, they are carried in the blood with a protein carrier because they are lipid-soluble and they don't want to dissolve in the water-based plasma. Once they are arriving at the target cell here, we're just going to follow the most common pathway. So we're going to cross the phospholipid bilayer of the plasma membrane, no problem here. We're going to follow along here, this arrow, and go directly into the nucleus. That is what I want you to know. So in the nucleus, so we're crossing here the phospholipid bilayers of the nuclear envelope, no problem there, free diffusion, and we're going to bind to a nuclear receptor. So once the hormone has bound its nuclear receptor, then together they become a transcription factor that binds to DNA and alters gene expression. And so here, then we're going to get an altered pattern of transcription, and that, of course, leads to a translation of whatever was transcribed here on the messenger RNA, and then we have our cellular response because we have these new proteins that are then causing the cell to do something. So here in steps, here is your hormone, lipid-soluble messenger bound to a protein carrier to travel in the blood. And then we are entering the cell freely by diffusion. No receptor necessary right here. So we're going to go into the cell by diffusion, going straight down into the nucleus. In the nucleus, we're going to find our nuclear receptor. We're going to bind to our nuclear receptor. And together, the hormone plus its nuclear receptor will then bind to DNA. And that's what you have right here. These are called hormone response elements. We had that in the lab. I was actually mentioned it was an HRE. These are hormone response elements in the short sequences of DNA where the complex of the hormone with its nuclear receptor will bind to and then alters the level of gene expression of the genes that are regulated by these hormone response elements. And so now here we have these genes, certain genes are being turned on or turned off, and so you have an altered pattern of transcription of messenger RNA, and that leads then to the various proteins by translation, and eventually we have these new proteins or altered pattern of proteins that then leads to the cellular response. Okay. Now let's take a look at some specific hormones and examples. We're going to use the amino hormones first in this category. So these will be amino acid derived hormones. Here we have melatonin, and that one helps with your circadian rhythms. It's a brain hormone. I'm always surprised that it's over the counter available. We don't really understand how this one works too well, but anyway. Catecholamines, these are very important for us, of course. We constantly talk about epinephrine, norepinephrine. I sort of lump these together. They're very related, and anyway, just usually I say epinephrine / norepinephrine and sort of grouping them together. And then dopamine is also an important catecholamine. They are derived from tyrosine. And then the thyroid hormones, T3 and T4, they also derive from tyrosine, but in this case it's two tyrosines together. So here is the amino acid tyrosine, unmodified, and you can tell that the catecholamines, the dopamine, the norepinephrine, epinephrine, they all look almost identical to the tyrosine. So small modifications that lead to these amino-derived hormones derived from the amino acid tyrosine. So dopamine, norepinephrine, and epinephrine, very, very related, very closely related. And then here we have our thyroid hormone. You can kind of tell on the tail end especially, it definitely looks like tyrosine, and we have another one of these ring structures that then give it the lipid properties that it has. So as soon as you add another one of these ring structures, now this is no longer water soluble, and you just have to carry it in the blood with a binding protein, and it follows the pattern of the other lipid-soluble messengers. But anyway, so here this would be tetraiodothyronine. So we have 4 iodines, 1, 2, 3, 4. So 4 iodines, and this is triiodothyronine, T3. So we have 3 iodines, 1, 2, 3, right here. And again, those are your 2 thyroid hormones. And if you remember from the lab, what we make mostly is T4, and then what's biologically active would be the T3 mostly. Okay, now we're going to take a quick look at endocrine reflux pathways. So here you follow this pattern here of a stimulus, sensor, input, a signal, and an integration. It's usually something in your central nervous system. Usually the hypothalamus is involved a lot of times, and then you get some output, targets, response, physiological action, and then usually negative feedback. So here, sort of a diagram that explains this kind of pathway. Here they're using low plasma concentration of calcium, and that will send a message to the parathyroid cells, and they need to release parathyroid hormone, which is carried in the blood and then acts on the target tissue to ultimately increase the plasma concentration of calcium. Once it has been achieved, if you need to send negative feedback to say, hey, we have enough calcium, we don't need to have any more parathyroid hormone. Or here is